Hello friends, today we will be discussing about digestion and absorption of carbohydrates, glucose transporters, ORS, renal glycosuria and lactose intolerance. Okay, so we'll start with digestion and absorption of carbohydrates. It's easy, just I'll write like an answer for each of these. We'll be covering these two together. Glutes usually MCQs, all these things usually MCQs is asked. The main questions that come from this part is digestion absorption of carbohydrates and lactose intolerance. Okay, so regarding digestion and absorption, of carbohydrates, it's easy. It is divided into two, as the name indicates, digestion and absorption. So in the digestion, so we'll write the answer in these headings. First is digestion, second is absorption, and you can write these three in the clinical application part. I have told you always, when you write an answer, it is better that you mention at the end under a heading clinical application. Whether it is any question, any subject, you can apply that clinical correlation or clinical application. So under that we will discuss these three. Okay, so regarding digestion and absorption, you can just giving an overview of both. Digestion is actually divided into digestion in mouth, digestion in stomach. You can write these headings. Intestine. Okay, these are the main headings in this, and intestine itself. Okay, there we'll discuss it there. And absorption again can be divided into facilitated diffusion, and active transport. Okay, these are the two main mechanisms related with absorption. Okay, active transport. So we'll start with discussion digestion first. So the first heading that you have to write with the answer is digestion. Okay, now speaking about digestion, for digesting in the diet we have what all things to digest. Starch is there, glycogen is there. Now what you have, other things like cellulose. If you are consuming plants or vegetarian diet, you will have cellulose also with that. Cellulose will be there. Then other things like glucose, fructose, lactose, part of milk, galactose and all, right? So here these three are the main monosaccharides. These are the three will be absorbed directly, but we have to learn the digestion has to happen for these things. Okay, cellulose we cannot digest, I already told you, but we can digest lactose and starch. Okay, glycogen discussion is not needed. The starch, lactose and lactose, how they are digested, we'll see. Okay, so the diet composed of these things. We usually eat these things, or the things that we usually eat has these components in them. Now the diet is this. Now coming to digestion, now what happens in the mouth? Yes, in the mouth we have an enzyme known as salivary amylase, produced by the salivary gland. Yes, salivary amylase is there. It acts only in the alpha 1,4 bones, or alpha 1,4 bones only. This enzyme acts. So, and it requires a pH more than 4 pH to be active. Okay, so whenever it goes below 4 or very acidic pH, it won't act. So that is how salivary amylase is. Now salivary amylase will act on starch that we have ingested in our food, and it will break down starch into its products like glucose, maltose, isomaltose will also be produced, and finally limit dextrin. Okay, limit dextrin is again a polymer product of starch when it is partially digested. And you know, in starch there are alpha 1,4 and alpha 1,6 bones, but only alpha 1,4 bones are broken by salivary amylase, that itself very partially. So now the whole of starch will not be digested, only partial digestion of starch will happen, and we have these products from the mouth itself. Yes, and this is the product known as limit dextrin. Okay, limit dextrin is also formed from partial starch digestion in the mouth. So these are the points of digestion you have to mention in the mouth, in the digestion phase that occurs in the mouth. Okay. Now the next thing that you are going to see is in the which phase it is in the stomach. Okay. So as you know, as you might have guessed it already, in stomach no digestion of carbohydrate happens because the pH is less than 2 or 2 to 4. You can say less than 4. Okay, less than 4. So it is a very acidic pH because who is there? Hydrochloric acid is there, which is produced by gastric cells of the stomach. Okay, gastric cells of the stomach. So the salivary amylase will get inhibited. The salivary amylase will be coming along with the food particles, digestive food particles into the stomach, but this salivary amylase will be inactivated because of the low pH, very low pH in the stomach. Okay, so that is a phase in stomach. Now the more important phase of digestion is happening in the intestine. Okay, so what happens in the intestine? Yes, what happens in the intestine? Okay, intestine we have two types of enzymes, or actually it is happening in with the help of pancreas and also with the help of intestine. Okay, intestinal juice and pancreatic juice. So these are the two juices that help in digestion in the intestinal phase. So in pancreatic juice we have pancreatic amylase, which again will act only in the alpha-1,4 linkages, because it again acts just like salivary amylase. The origin is only different. This is pancreatic, produced from the the exocrine pancreas or serous acini cells. You might have learned in histology. So pancreas, exocrine part of the pancreas actually produces pancreatic amylase, again alpha-1,4, just like salivary amylase. And the main intestinal juice produced by the crypts of Lieberkühn, which you might have studied in histology again, crypts of Lieberkühn. Okay, this produces what all things? These have enzymes known as disaccharidases. Okay, disaccharidases means these are the ACE means the enzyme, right? It acts on disaccharides. I'll tell you. Now, before going to intestinal juice, before we will cover the pancreatic juice, nothing, very simple. Already lot of the food has been digested into, right? Write it here. The food from the mouth will be like this. Partial starch digestion would have happened in the mouth, and we have little starch remaining because it is partially only digested. Glucose, maltose, which is a disaccharide, isomaltose, another disaccharide, and also limit dextrin, right? Limit dextrin. And also if you have had milk or milk products, this also will be there in the food that reaches the intestine. This will not be digested by salivary amylase in the mouth because it is a disaccharide and it has no action on disaccharide. So lactose and partial starch and maybe glucose and other things will also reach the intestine. So what will pancreatic amylase do to this food that reaches the intestine? It will break this starch fully. Okay, starch when fully digested will become glucose. All this limit dextrin and all will be fully again digested. Limit dextrin cannot be digested because it is in the alpha-1,6 linkages. Because it has alpha-1,6 linkages, pancreatic amylase also cannot act on limit dextrin. So again the rest of the starch will be digested, and again these products will be formed. Glucose, maltose, isomaltose, and limit dextrin again will be formed. Limit dextrin. Okay, so what happens in the intestine with the help of the pancreatic juice is that the complete digestion of starch happens, and we have glucose, maltose, isomaltose, and limit dextrin. Okay. Yes. So that is the action of pancreatic amylase. And remember, in the intestine, it has a basic pH, right? It has a basic pH because intestinal cells or juice is actually basic in nature. Whatever is produced from the crypts of Lieberkühn is basic in nature compared to stomach where it is acidic environment is there. So we have basic pH in the intestine, that is, so pancreatic amylase is active only in the basic pH. So that is a pancreatic thing in intestine. Now talking about intestinal juice, I told you disaccharidases is contained in the juice produced by the crypts of Lieberkühn. Now these disaccharidases, okay, I think you already know there are villi in the intestine, right? Especially in the small intestine, we have small villi here. So these disaccharides are actually situated on the villi, okay, on the villi in the intestine, microvilli in the intestine. So whom do these disaccharidases act on? Disaccharidases can act on these food products, okay. Let me write it again. Glucose is there, fructose can also be there if you have consumed fruits and all. So fructose can also be there, that you can add to the, is already there. Fructose can also be part of your diet. Lactose is there, and we have, what you have, maltose, isomaltose, and limit dextrin, okay. Now focus here, these are the disaccharidases, enzymes that are located on the microvilli, okay, on the brush border epithelium. So I told you disaccharidases are there. So what are the disaccharidases which are present in the brush border epithelium? One is maltase, okay, maltase, which will obviously digest maltose into 2 glucoses. Maltase will be present. And isomaltase will also be present. Isomaltase. Before that, rather than that, first we'll learn about lactase, okay. We have lactase here, okay, maltase, lactase, and also isomaltase, okay. Actually it is existing as a complex, okay, sucrase isomaltase complex. Yes, and in the diet you can have sucrose also. I forgot to mention here, because sucrose is a common sugar. It can be present in the foods you eat. Whatever you consume can have common sugar in it, sucrose. That sucrose will also undergo digestion only in the intestinal part. So we have 3 main enzymes in the brush border epithelium. One is sucrase isomaltase complex, one is maltase enzyme, and the other one is lactase enzyme. These are the main enzymes. Now maltase will split maltose into 2 glucose molecules, okay, 2 glucose molecules. I have already taught you about the linkages present in a lot of the disaccharides in the carbohydrate chemistry class. Just go back and refer if you have any doubts. So maltase will break maltose into 2 glucose. Lactase will obviously break lactose into glucose and galactose, okay, glucose and galactose. Now sucrase isomaltase is interesting. Because it has sucrase in that complex, sucrose will be split into glucose and fructose, okay, glucose and fructose. Now isomaltase, just focus here, isomaltase, it is part of this complex, sucrase isomaltase complex, but this isomaltase will act on isomaltose and limit dextrin. And remember, in this whole digestion that we discussed, only isomaltase will have alpha 1,6 activity, okay. All the amylases, all the other things will have only alpha 1,4 and other linkage breaking properties, but alpha 1,6 breaking property is mostly by isomaltase. So that is why it is able to break isomaltose further into glucose and glucose. Again, isomaltose will be split into glucose and glucose. Limit dextrin again will be further, the polymer shape will be lost and will be digested into component glucose molecules. So this is how digestion of all the food that you have, that includes starch, glycogen, cellulose, glucose, fructose, lactose, galactose, and sucrose happens, okay. So it has the stomach part. The digestion has a stomach phase, mouth phase, stomach phase, and intestinal phase. Mouth phase, we have salivary amylase, where partial digestion of starch happens. Rest of the starch is digested by the pancreatic amylase when it reaches the intestine. Now, after that, we have the stomach phase, where nothing happens because of the low pH provided by the HCl. And finally, we have this intestinal phase, where it is divided into pancreatic juice and intestinal juice. Pancreatic juice has pancreatic amylase, again only action in the alpha 1,4 direction linkages. And yes, and coming to the intestinal part, where the main enzymes that are produced from the crypts of Lieberkühn are disaccharidases. Disaccharidases are mainly lactase, maltase, sucrase, isomaltase complex. Okay, all these are situated in the intestinal brush border. Okay, so this intestinal brush border things can split all these, and finally the products ready for absorption will be, which all are the products ready for absorption? It can be glucose, galactose, and fructose. Okay, these are the products that we have in the intestine now ready for absorption. So we'll see the the absorption phase now. So the first heading is finished, and subheadings also. So digestion is finished. Now we'll go to absorption. Okay. Okay, you have so the next phase that is going to start is absorption. Okay. Now, absorption of carbohydrate, when you talk about absorption, I told you there are two kinds of diffusion. One is facilitated, two kinds of transport. One is facilitated diffusion, which has a ping pong mechanism. You might have read this in your book, ping pong mechanism. And there is another type of transport, which is ATP using transport known as active transport. Okay. So these are the two main mechanisms by which absorption happens of these three molecules, that is galactose, fructose, and glucose. Okay. Sorry, we'll write this in this order: galactose, glucose, and fructose. Okay. Okay. So why I wrote in this order is the absorption of galactose is more than glucose. The speed of absorption. Okay, speed of absorption in the intestine. You can maybe remember or mention in your answer also. The absorption speed, when the body prefers galactose to be absorbed very fast, and then it goes for glucose, and the next speedy absorption is by fructose. This is the speed of absorption, descending order of speed of absorption. Okay, galactose, glucose, and fructose. And moving on to the details of facilitated diffusion, we have ping pong mechanism. So first we will cover facilitated diffusion. Just for an overview, I will tell you. Facilitated diffusion, will talk about the glucose transporters, GLUTs. Okay, glucose transporters, and active transporters mainly by sodium dependent glucose transporters. Okay, SGLTs. So about GLUTs we will learn first. Okay, now facilitated diffusion means it requires a protein. It requires a protein for transport. Requires a protein for transport of glucose or whatever molecule across to the intestine. And the next point in this, it will take the mechanism that happens in facilitated diffusion is the ping pong mechanism. And what else? Yes, so we'll start discussion of the GLUTs. GLUTs are the glucose transporters which use the mechanism of facilitated diffusion to transport glucose, fructose, and galactose into the small intestinal cell. Okay, so I hope you know the main, that is, this is the lumen of the intestine. If this is the lumen of the intestine, small intestinal cell will be opening to this side, right, to the facing the lumen. This is the luminal side of the small intestinal cell, and this is the basolateral side or the serosal side of the intestine. So because there are glucose, fructose and galactose now ready for absorption, these three should be absorbed into the cell first, and then it goes to the blood, okay, for distribution to various tissues and cells. So first this absorption should happen, and glutes actually help for these kind of absorption, glucose or these monosaccharide absorption into the cells. So we'll see about glutes. We'll write this list here. Okay, the one will be a transporter's name, transporter, and then we have the sites it is seen, sites these things are seen, and also the function of each of them. Okay, I think we'll need this whole part too for the discussion. So we have transporter, sites each transporter is seen, and the functions of each transporters. Okay, so first thing we have is, who will be the first guy? Okay, GLUT 1. Okay, GLUT 1. Okay, we are talking about absorption. In that it is divided into facilitated and active. Now we are talking about glutes under, or the facilitated diffusion under absorption. Okay, so facilitated diffusion has these glucose transporters helping them: GLUT 2, GLUT 3, GLUT 4 and GLUT 5. Okay, these are the main 5 glutes that you have to know. Remembering the site is easy. See, the main sites only you have to remember. GLUT 1 is actually, GLUT 1 is, we are part of, GLUT 1 is actually seen in 2 sites. One is brain and RBC, and also some books have mentioned placenta, kidney, colon also. Okay, not important, you just have to remember mainly brain and RBC. Okay, so why is GLUT 1 found in the brain and RBC? And these GLUT 1 will have low Km also. Low Km means high affinity, right? So why do brain and RBC require glucose so much, or the glucose transport in these distributed in these is GLUT 1, and why does it show low Km or high affinity? Because these two things require glucose so much for their survival. They cannot depend on other sources, mainly they are dependent on glucose. Especially in RBC, you know, there is no mitochondria, so the only way to generate glucose energy is for glycolysis. So anyway, RBC needs it for its survival. And brain also, the primary component, primary fuel for its functioning is glucose. So that is why GLUT 1 is distributed mainly in brain and RBCs. The function of GLUT 1 there is basal glucose uptake. Okay, basal glucose uptake, or simply glucose uptake you can say. Basal glucose uptake. Okay. Now talking about GLUT 2. Okay, GLUT 2. GLUT 2 is very important because it is found in 4 areas mainly. One is the serosal or basolateral surface of the intestine. Serosal surface of intestine. Okay, and we have the beta cells. It is also seen in the beta cells of pancreas, which produce insulin. Okay, yes, and we have serosal surface of intestine, beta cells of pancreas. Yes, liver sinusoidal cells for absorption of glucose into liver sinusoidal cells, and basolateral membrane of PCT in kidney. Okay, so this is important. This is where GLUT 2 is found. Okay, serosal surface of intestine, beta cells of pancreas, liver sinusoidal cells, and basolateral membrane of PCT in kidney. This is also having yes, no, not about this. So this is these are the sites where GLUT 2 is seen. And about the functions, Yes. So just as I said, for GLUT1, GLUT1, GLUT3, and GLUT5 has actually low Km, okay, and GLUT2 has high Km. That means low affinity. So only only when the blood glucose level is very high, only GLUT2 will open for the cell and make the glucose enter the cell. Okay, GLUT2 will open up only in high, I mean when the blood glucose level is very high, because it has only low Km, that sorry, high Km, so that means low affinity. Okay, so this has high Km. Okay, so that means low affinity. So only when the blood glucose level, so you can think like cirrhosis of intestine, beta cells of pancreas. Beta cells of pancreas, if you think about that, only when the blood glucose level reaches a particular point, only it should secrete insulin. So for that, the GLUT2 has been placed in beta cells of pancreas. And also for the other two, liver sinusoidal cells, because only if there is high glucose in your blood, only the liver should allow into the glucose into those cells for storing it as glycogen by making glycogen from glucose. So that is the logic behind having GLUT2 in these areas. Okay, now yes, so that is the function that I've just told. Okay, intestine, pancreas, and sinusoidal cells will have this because it has high affinity, high Km, and therefore low affinity. So these things will only open when the level of glucose is very high, very high, not so much. Then when it is crossing a particular limit, only these things will open the channel and let the glucose inside. Now talking about GLUT3. Okay, 3 looks like a neuron, right? In the brain you can see a neuron, or it stands for neuron. So usually it is seen in the neurons. Okay, mainly in the neuron. GLUT3 is mainly part of the neurons. Okay, again it is only glucose uptake. The main function is glucose uptake. Okay, now coming to GLUT4. Okay, GLUT4 is a special kind of mechanism, because this is the only transporter of the all 5 transporters which is insulin dependent. I will tell you. So all these GLUT4 is actually spread over skeletal muscles, adipose tissue, heart tissue, and all. Okay, so the distribution is in these tissues. GLUT4 is distributed in the heart tissue, skeletal muscle, and adipose tissue. Okay, adipose tissue. So these are almost all over the body who is being attached to these membranes or acting as a gate for glucose. So it is present in the heart, skeletal muscle, and adipose tissue. Now what is the function for GLUT4? It is again same, but this time it is insulin dependent glucose uptake. Okay, insulin dependent glucose uptake. That means only when insulin comes and tells the transporter to open, GLUT4 to open, only then it will open. Not like these other guys. These other guys are not obeying insulin's order. Only GLUT4 will be obeying insulin's order to open the channel and let the glucose into the cell. So insulin dependent glucose transporter is this. Okay. Now GLUT5 is the only fructose transporter we have. Even though it is known as a fructose transporter, it can also transport glucose and galactose in small amounts. Even if I have mentioned only glucose uptake in these other GLUTs, it also transfers some amount of galactose in it. But the only unique transporter or only exclusive transporter for fructose is only GLUT5. Because it is a fructose transporter, you can imagine which all places would require fructose in our body. Okay, yes, we need small intestine. Okay, for some purpose it needs fructose, but the main areas are sperm. Okay, sperm needs fructose in it, so testis also needs it for synthesis of new active sperms. Sperm actually depends on this fructose that is present in our body for its motility. Okay, so it is important for the sperm to have fructose transporter known as GLUT5. So as I told, the main transporter, the function is it acts as a fructose transporter. Okay, right, fructose transporter. So these are the main glutes that you have to study under facilitated diffusion. Okay, that comes under absorption. Okay, facilitated diffusion, these are the main glutes, and GLUT 1, 3 and 5 are supposed to have low Km, so they have high affinity for glucose. So even with low levels of glucose itself, it will be taken into brain and RBC and other areas and all. Okay, but keep in mind GLUT 2 has a high Km value, so that means only it has low affinity, so only when the glucose crosses a particular limit only these things will take the glucose inside. And GLUT 3, as a number indicates, it is for neurons, part of brain which is neurons. It is also involved in glucose uptake. GLUT 4 is the most widely spread GLUT present in our body. It is present in adipose tissue, heart tissue and skeletal muscle. It is the only one insulin dependent one, so it is only obeying insulin's orders. Okay, GLUT 5 is the only fructose transporter present and is seen in small intestine, sperms and testes. So this is about glutes. Okay, now even if this question is not asked for a detailed answer, you can expect MCQs coming from these. That's why I elaborated somewhat. So thereby we finished the facilitated diffusion part of absorption. Now we'll see the active diffusion part. Now for the absorption, the next active transport part is active transport part. Active transport part, yes. Now coming to active transport, we have two kinds of transporters, SGLT1 and SGLT2. Okay, yes, and SGLT1 is mainly present in the intestine. Okay, and SGLT2 is mainly present in the kidney. Okay, yes. Now regarding this active transport, we have to understand how this transport happens. So for that we will see a intestinal cell. Okay, intestinal cell, this is the intestinal cell with brush border epithelium, and this is the intestine where the lumen is there, right? The lumen will be all of the lumen will be made up of cells like these cells, intestinal epithelial cell, and the part where it opens to the blood is known as the serosal part or the basolateral part. Okay, so you have blood vessel here. So whatever food comes through the small intestine tunnel, it will be absorbed by the digested particles, like the final products like glucose, galactose and fructose will be absorbed into the epithelial cell and then sent to the blood through the blood vessel. So what I am trying to show here is SGLT1 is usually seen in the intestine and it is seen in the which area? In the luminal part of the cell. This is the basolateral part, this is the luminal part. Luminal part has this thing known as SGLT1. Okay, and the luminal, not luminal, the basolateral part will have transporters known as GLUT2. Okay, I told you it is present in the basolateral or the serosal part of the intestinal cell. So GLUT2 is here, and also another this I have to show. Usually in the serosal part, this pump is also present, which is sodium potassium pump, which will pump out 3 sodium and at the same time it pumps in 2 potassium. Sodium potassium ATPase pump, it uses ATP for the activity. So this is a ATP pump. Okay, so sodium potassium ATPase. So what I am coming to tell is, we are talking about absorption and that too about active transport. Okay, so that means if glucose is there or galactose is there, glucose and galactose both uses this to come inside. it will come inside like this. But because it is a co-transporter, sodium dependent glucose transporter, so that means it should also attract what inside inwards? Sodium. So because it happens together, whenever glucose has to be absorbed, sodium will also be absorbed. So that is an example for co-transport. Now another question that is usually asked in the exam is, when this SGLT1 is actually what kind of transport? Even though we are learning it as it under active transport, this is actually secondary active transport. So what do you mean by? So SGLT1 is involved in secondary active transport. Okay, it is not directly using the energy. I'll explain why it is called a secondary active transport. Okay, see, in the serosal side of the cell, a pump is actually working utilizing ATP every time and pumping 3 sodium outside and the 2 potassium is taken inside, right? So this is an active process because it is utilizing ATP. Now because the sodium is pushed outside, there will be a sodium deficiency here, right? The sodium deficiency will occur inside the cell. This sodium deficiency is what actually drives the sodium along with the glucose into the cell. So actually when you think of that, sodium is not taken by directly using the ATP, but ATP usage happens here and a sodium deficiency is created. That deficiency drives the sodium inside together with glucose. So that is why it is most commonly known as a secondary active transport. Okay, so this is a wonderful example for secondary active transport. Can you tell me the sodium and glucose coming together, where is this feature utilized for treatment of patients? Yes, you said it right, it is used in ORS. Okay, ORS, if you look at the contents of the ORS, you will see glucose and salt and sodium along with it. Why? Because only if you give, as ORS is given in when the patient feels dehydrated, when the electrolytes and the things are lost in diarrhea or dehydration or some vomiting and all, we can give ORS so that the electrolyte status and the glucose status gets restored. So while using ORS, you can get both glucose and sodium. So the electrolyte balance of sodium levels can be managed and also glucose will also be absorbed into the body. So that is why SGLT1 is a mechanism utilized mostly in the ORS. So that you can mention in the clinical application part also. So this is about SGLT1. Now talking about renal glycosuria. Yes, ORS also we mentioned. Now talking about renal glycosuria, what happens? See, there is a renal, do you know the concept of renal threshold for glucose? Yes, what is the renal threshold for glucose? See, the renal threshold for glucose is around 175 to 180 milligram per deciliter. Okay, understood? Now why this value is important, or why is it known as the renal threshold for glucose? That means in most of the normal people, like who are not suffering from diabetes or anything, usually and even people with diabetes also, only when the blood glucose level, the glucose level in the blood, only if it crosses this level 175 to 180, only then the glucose starts to appear in the urine. It is a normal thing to find glucose in the urine when the blood glucose level crosses this. Okay, and that level above which urine glucose will be start coming is known as the renal threshold for glucose. That is why this value is known like this. Now in some special circumstances, what happens is this renal threshold for glucose will be lowered. So what happens when the threshold is lowered? Even with small amounts of glucose in the blood also, who will start to appear in the urine? Glucose will start appearing in the urine. Understood? Usually the renal threshold for glucose is this. Only when the blood glucose level crosses this, only glucose starts appearing in the urine, should start appearing in the urine. But in some cases, in some patients, this threshold itself is lowered. Threshold lowered means it can become maybe 110, 100 or something like that. So even with normal levels of glucose in those patients, blood levels in those patients, sugar starts coming very early. Such cases are known as renal glycosuria. Okay, renal glycosuria. Okay, this is the renal glycosuria part. So renal glycosuria means the lowering of renal threshold for glucose. Now why does that happen? Why did I mention this during this? Because renal glycosuria can be either congenital or acquired. As the name indicates, if you have renal glycosuria due to some genetic defect or from birth itself, it is known as congenital. Acquired renal glycosuria is also there. Congenital renal glycosuria, what happens actually is this SGLT transporter gets defective. In such patients, SGLT2 transporter gets defective and kidney absorption of glucose will stop. So that's why urine will be losing glucose at the very low renal threshold itself. So that is known as renal glycosuria, congenital type. Acquired type can happen when in kidney diseases. Again, in kidney diseases like diabetic nephropathy or any kind of hypertensive nephropathy, those kind of kidney issues also, what will happen? This renal threshold will come down due to kidney damage. Then also we can find renal glycosuria. That is why it is known as acquired. Due to disease it comes. If it at birth itself, if you have, it is known as congenital. It is usually due to SGLT2 defect in the kidney. Okay, so that is about transport. We have covered digestion and transport, and also the main two things that we discussed is ORS and renal glycosuria. We'll talk about lactose intolerance now. Lactose intolerance, we have what is the main issue? So you can write it like definition. Okay, the heading should be definition first. It is the lack of enzyme lactase. Okay, lack of enzyme lactase is there, so not able to digest. That is the simple definition, not able to digest lactose. Okay, this is the main issue, lactose. Now what is the main biochemical problem faced here or during digestion? What happens? Because lactase is not there, because lactase is not there, what happens? The lactose in the food that we digest or that we which reaches the intestine will be acted upon by intestinal bacteria, which cannot digest it, but it ferments the lactose. So lactose intestinal bacteria will act and it will ferment the lactose here, and lactose on fermentation will give out hydrogen, methane and carbon dioxide. Okay, so when you give out these gases, and also some lactic acid will also be produced. Lactic acid, short chain fatty acids and all will be produced along with the fermentation of lactose. But the main gases are formed are hydrogen, methane and carbon dioxide. So what happens to these gases is this causes because lactose is so there are many clinical features. Okay, you can write the next heading as clinical features. Now lactose being very osmotically active, it will attract water in the intestine, so diarrhea will be there. Diarrhea will be there because lactose is osmotically active. It will draw water. Diarrhea can happen, nausea, vomiting and all can happen. And one more main symptom is, because these gases are there, flatulence is a main problem. Okay, you will have a gassy stomach because of these gases, and you will be losing gas through your anal opening. So that is known as flatulence. So flatulence, diarrhea, nausea and vomiting are the symptoms which is usually accompanying lactose intolerance, because you are intolerant to lactose. Your body cannot handle lactose. Now after writing definition, biochemical issue and this clinical features, maybe before that only you can add another heading. Before biochemical things, you can add another heading known as what the types of lactose intolerance. Okay, types of lactose intolerance. Okay, types of lactose intolerance are: one is congenital, which is present from the birth. Another one is primary lactose intolerance. Another one is secondary lactose intolerance, like that. So the main three ones are congenital, primary and secondary. Now in congenital thing, what happens is the babies are born without with the complete deficiency of the enzyme. Complete deficiency of lactase. Okay, lactase, complete deficiency from because of a genetic defect, they are not having lactase itself. And primary is due to the born child will have lactase enzyme with it in the stomach of the baby, but in the intestine of the baby. But as age progresses, after 4 years or after 2 to 4 years, what happens is the lactase enzyme level will gradually decline. So that is what is known as primary lactose intolerance. Now, secondary lactose intolerance happens due to some diseases. If you have a disease like Crohn's disease, celiac sprue, or gastroenteritis, I'll write those names. Gastroenteritis is your common food poisoning can happen due to gastroenteritis. Okay, inflammation of the intestine and the stomach because of bad food that you have eaten. The germs in that and the microbes in that will cause gastroenteritis. Another disease is known as celiac sprue. Again, we are losing a lot of food because of malabsorption. Celiac sprue is there, and also another disease known as Crohn's disease. Okay, Crohn's disease is a disease that affects the intestine. In these cases also, the intestine can itself be damaged. So you already know lactase is an enzyme which is a disaccharidase. Disaccharidases are present in the intestinal brush border. So if you have diseases like this, the intestinal brush border will itself be damaged, and you will lose lactase enzyme, leading to secondary lactose intolerance. Okay, so these are the types of lactose intolerances I wanted to mention. So you can write the answer like this: definition, types of lactose intolerances, biochemical defect, and clinical features, and finally about the treatment. Okay, treatment is easy. If you have intolerance to lactose, avoid lactose based diet, like dairy products, milk and all can be avoided. And we have some other treatment options also these days. Indians don't generally have lactose intolerance, usually found in the Western population. So the treatment part for lactose intolerance is you have to decrease or avoid milk and milk based products. Okay, avoid dairy or milk and dairy products. Okay, and sometimes you can have lactase. These days you have lactase containing capsules. Okay, lactase containing capsules that you can have, and it will go to the intestine and it will create a lactase rich environment in your intestine. Another option is you can have curd included in your diet. Curd in your diet, because curd has lactobacillus in it, an enzyme known as lactobacillus. This lactobacillus can actually, this lactobacillus bacteria has which enzyme? Lactase enzyme with it. So curd can help in this disease. Another one will be if you slowly increase your lactose consumption, little by little. If you have lactose daily, not in a like a big bowl of milk or something, small amounts of lactose if you introduce to your stomach, lactose quantity, okay, in your diet, slowly increase lactose quantity in your diet, that will lead to lactase induction or lactase enzyme increasing in your intestine. This is because lactase is a inducible enzyme. That means as you give lactose, the body will try to create more and more lactase in the due course of time. So this is because this is a inducible enzyme, you can introduce lactose in small quantities. So these are the treatment options available nowadays. So I think lactose intolerance, if it is asked, is usually asked. So you can cover the answer in these headings. Okay, thank you.